So as I mentioned, that the levels of organization are starting with chemicals and then with cells, and the cells will form tissues. And so today we are going to deal with cells. We are going to have 2 lectures on the cells. This is the first lecture today. And the cell is the basic living structural and functional unit of the body. The cell has a membrane surrounding it, a cell membrane. Its main function is to regulate the passage of material or inflow and outflow between what is present inside the cell, intracellular compartment, and what is present outside the cell, which is called extracellular compartment. So this is the cell membrane. It separates, it protects, and regulates the flow of structures. And inside the cell, there is a central region usually, which is called the nucleus. And this nucleus contains the genetic material that control the activities of the cell. Between the nucleus and the cell membrane is the cytoplasm. And the cytoplasm contains several small structures, which are called the organelles. We're going to study these organelles next week. This is a typical cell. It probably contains all the possible organelles, but you will not expect that all the cells in the body contain all these organelles, because these organelles, each of these organelles has a certain function. And each of these organelles will do its function separate from the others. There's compartmentalization. The functions of the cells are done in different compartments, in different organelles. So depending on the function of that cell, the organelles will predominate. We will talk about that in details later on. But this is just an imaginary typical cell that contains all the possible organelles. So again, I repeat that the cell, which is the main structural and functional unit of the body, basic structural and functional part of the body, contains a cell, has a cell membrane, surrounding cell membrane. There is a nucleus, and everything between the nucleus and the cell membrane is the cytoplasm. The cell membrane is flexible, but it is a strong one. We will see how this happens. It surrounds the cytoplasm, and the cytoplasm can be divided into 2 parts. The fluid part of the cytoplasm is called the cytosol, and the organelles, which are smaller organs, so organelles, subcellular structures. They do not constitute a living structure by themselves, because the smallest living structure is the cell. So what is the function of the cell membrane? We mentioned that it acts as a barrier that separates the external environment from the internal environment, the extracellular from the intracellular. And it also controls the flow of substances, either inside the cell or outside the cell, because many of the cells, they either secrete or excrete. By excretion means that they will get rid of the waste products, and by secretion means that they produce substances that are important for the function of the tissue or the organ or the body itself in general. So the cells, some of the cells secrete hormones that will travel to other parts of the body and exert their effect. Some other cells will produce and secrete enzymes that act, like, for example, the digestive enzymes that usually act locally. And of course, all the cells, they will excrete, they will get rid of the waste products. So this is the difference between secretion and excretion. The cell membrane also helps to identify the cell to other cells. So, for example, the immune system can recognize foreign cells or can recognize bacteria as they are harmful substances and will attack them. So they are used as markers for that cell, and it's also used for intercellular signaling, means signals between the cells, communication with other cells. This is very important actually during embryological development, or it's also important during regeneration of tissues, so that the cells, they have some sort of communication between them and they can organize themselves in the way that they should be organized. So the cell membrane is very important. That's why it has like some sort of complicated structure. This is the structure of the cell membrane. It consists of molecules, as we mentioned previously. This is the lowest level of organization. And these molecules are lipids, proteins, and carbohydrates. There's one method of describing the cell membrane. This is called the fluid mosaic model for the cell membrane. By fluid, as you can see here, that these are the blue structures here. They are lipids. And you can see that the lipid material is in the form of a double layer, lipid bilayer, back to back, and they are floating like in a sea. So these molecules of lipids are floating in a sea. So the cell membrane is very flexible, and in addition to that, you can see that within this sea of lipid molecules, there are other large molecules here. These are proteins. Some of these proteins, they are just floating with the lipids, so they are like icebergs here. But others, they are anchored, they are attached in place like islands in a sea. And some of the proteins, as you can see, are either attached to one surface of the cell membrane, or they might extend the entire thickness of the cell membrane, and they are called transmembrane proteins. Okay? So there is a sea of lipids. Proteins are floating. Some of them are floating like icebergs. Some of them are anchored in place like islands. And these proteins, as we will see in a moment, they are very important. One of their functions is like they act as gatekeepers. They allow the passage of substances that are not allowed to pass through the lipid bilayer. And then the third structure is carbohydrates. As you can see, these carbohydrates, the dark blue structures, dark blue chains here, some of them are attached to proteins, and some of them are attached to lipids, like this one. This one's attached to lipid, this one's attached to protein. So the ones that are attached to proteins are called glycoproteins. Glyco means like from glucose, sugar. And the others which are attached to the lipids are called the glycolipids. Glycoproteins and glycolipids together, they will constitute what we call the glycocalyx, which is like the glaze, the sugar coat of the cell membrane. So the cell membrane contains three basic types of molecules, main molecules, the lipid, the protein, and the sugar. And this is the way that they are organized. The lipids are in the form of a bilayer, it's like a sea, and the other molecules of protein are floating in that sea or attached to the sea, and there are other molecules of carbohydrates either attached to the protein or attached to the lipid, and they form the glaze, the sugar coat of the cell. The lipids are in three parts. They are not all of them the blue structures here. These are only the phospholipids, and actually they are the main part of the lipids. But there are other types of lipids, like the cholesterol here, and the glycolipids, which we mentioned, the lipids that are attached to glucose molecules. So the phospholipids, as you can see, that they have polar heads and non-polar tails. The heads are also termed as hydrophilic, and the tails are hydrophobic. So that's why the heads are always facing the part which contains water, because they are hydrophilic. They like the water, unlike the tails, which are hydrophobic. They are afraid of the water, so they turn away from the water. So here, the outer part of the cell membrane, the heads are hydrophilic, and they face the extracellular compartment, the extracellular fluid, and from the inside, the heads are facing the intracellular compartment, the intracellular fluid, but the tails are sandwiched in between. They are hydrophobic. Carbohydrates, as I mentioned, are attached to some of the lipids, and these are called glycolipids. This is part of the glycocalyx. Cholesterol can be attached. They are smaller molecules. They are much less than the phospholipids, and they are attached either to the outer or to the inner layer of the cell membrane, either to the outer or to the inner layer of the cell membrane. They provide flexibility and strength at the same time. So that's how the cell membrane is flexible and strong at the same time. And having these molecules just floating like that, it means that part of the cell membrane can be pinched out without affecting, without causing an opening in the wall of the cell. Just can be just pinched out, and the remaining cells will organize themselves back again. This is what we are going to see in endocytosis in a moment. Or a vesicle can attach to the cell membrane, fuse with the cell membrane without making any disturbance, because it is flexible and it is floating. The proteins, the second constituent of the cell membrane, are either integral proteins or peripheral proteins or glycoproteins. The integral proteins, as the name indicates, they are firmly embedded in the membrane, as you can see them here. This is an integral protein. And they either extend through and through the cell membrane, which are called transmembrane proteins, as the name indicates, transmembrane, through and through. And some of them are small proteins, are either attached to the outer layer or the inner layer of the cell membrane. The peripheral proteins, on the other hand, they can be either attached to the outer or inner surface of the cell membrane. This is a peripheral protein. They are not firmly attached. They are lightly attached, not as the integral proteins, which are firmly attached. And then we have the glycoproteins. Whenever there is sugar molecule attached to the protein, it's a glycoprotein. Same principle as the glycolipid, as I just mentioned. And these two, glycolipids and glycoproteins, they produce what we call the glycocalyx, the glaze, the sugar glaze. And it's only related to the outer part of the cell membrane. There is no glycocalyx on the side of the cell membrane that faces the cytoplasm, only on the side of the cell membrane that faces the extracellular fluid. This glycocalyx is important for cell recognition. So again, it acts like a molecular signature. We mentioned that this is one of the main functions of the cell membrane. So the glycocalyx here is important in that. And it creates stickiness. It's sugary, so it creates stickiness. So in some tissues, it will enable the cells to adhere to each other because it's sticky. And it protects some other cells from being digested by digestive enzymes. And it also attracts water to it. So this makes the wall of the cell slippery. Like, for example, the red blood corpuscles or red blood cells, when they pass through tiny small vessels, which are called capillaries, they need to fold themselves and to slip in between. So having a sugar coat, the sugar coat attracting water, it will make them slippery and easy to navigate and pass through that narrow blood vessel. On the other hand, the attraction of water will prevent from dehydration, like for example in other tissues, like in the respiratory system or the gastrointestinal system. Having this coat of sugar, which will be coated with water because it attracts water, will prevent the tissue from getting dried, because in the GI, gastrointestinal, and the respiratory system, in certain places, the tissues are exposed to the exterior, so to the outside. So there's air passing in, and this will cause dryness. Having this sugar coat will attract water and make the coat wet and prevents dryness. So going back to other functions of the cell membrane is they form channels or pores, as you can see them here, for passage of certain substances. Certain substances cannot pass through the lipid bilayer, especially the polar substances like electrolytes, like calcium, sodium, potassium. They cannot pass through the lipid bilayer for chemical reasons, so they have to pass through, find another way to pass through, and so they pass through the channels or pores that are created by the membrane proteins. Some of these proteins that are present in the membrane not only act as channels, but also they act as what we call transporter or carrier proteins. Some molecules, like for example the glucose, which cannot pass through the lipid bilayer, has to pass through the proteins, but the shape of the molecule cannot accommodate itself through the pore. So they have to be attached to another protein, like this one here, which will change the shape of the original molecule and make it susceptible to pass through the narrow pore. So this is another kind of protein. Another function of the protein in the cell membrane is that it acts as a transporter protein. Or they may act as receptor proteins. Substances attached to the cell change the function of the cell. These substances, these specific substances that attach to the receptors, are called the ligands, and these receptors are made of proteins. So the proteins, they could either form pores in the membrane for the transport, or they could be transporter proteins by themselves that change the shape of the substance to be transported in order to accommodate itself into the pore, or they act as receptors so that ligands will attach to them and change the function of the cell. Also, the proteins are markers, cell identity, same as glycocalyx, and some of them act as linkers. They anchor proteins of other cells, or they might anchor proteins that are present in the filaments. Filaments are a kind of organelles that we are going to talk about next week when we talk about the organelles of the cell. So they anchor these filaments, which are present inside the cell. These filaments play an important role in changing the shape of the cell, as we will see that later. Some of the proteins, they act as enzymes. The enzymes, they speed the chemical reactions in certain places. So these are, in general, the functions of the cell membrane and the proteins in specific in the cell membrane. The cell membrane, as I have just mentioned, has selective permeability. As I mentioned that, for example, ions, calcium, sodium, potassium, they cannot pass through the lipid bilayer. They have to pass through specific pores. But on the other hand, other molecules like carbon dioxide, like oxygen, hydrogen, like steroids, they can pass through the lipid bilayer. Okay? So there is, you can see here that there is a selective permeability in the passage of structures. Not all the structures can readily pass. They have to select the place where they pass through. And if the molecules are big molecules, large molecules, then there's another way of passage through the cell. They have to be engulfed, they have to be taken in, and this is what we call endocytosis, or pushed out by the process of exocytosis, as I have just mentioned. The transport across the plasma membrane could be either passive or active transport. So in a passive transport, as the name indicates, there is no need to use energy, and so it is done passively. It is just done passively, and usually it is done down the concentration gradient of the substance. The other type of transport is active transport. And active transport, the substances will move against their concentration gradient, and in this case you need some energy. The ready source of energy that is present in the cell, the molecule is called ATP, adenosine triphosphate, and this is hydrolyzed or degraded into ADP, adenosine diphosphate. And this results in the production of energy. Energy is going to transport the material through the cell membrane against the concentration gradient. So active transport could be either diffusion through the lipid bilayer, for example, the simple diffusion of oxygen and carbon dioxide, for example, the wall of the alveoli of the lung. Oxygen will diffuse from the alveolus itself, from the space, into the bloodstream, because there is high concentration in the alveolus, low concentration in the blood. And on the other hand, carbon dioxide will pass from the blood to the center of the alveolus because there's high concentration of carbon dioxide in the blood coming or arriving to the lung. Yes?
So if exocytosis is when it's being pushed out of the cell,
Mm-hmm.
would that always be active transport more often than not?
Exocytosis, yes, it will be. It will need. It usually requires energy. Yes,
Okay.
but now I'm talking about diffusion. I will come to exocytosis in a moment. So diffusion doesn't require energy, and it is usually down the concentration gradient of substances. So as I mentioned, some of the substances, they can diffuse through the cell membrane, but they cannot pass through the lipid bilayer. They have to pass through a channel. The channel is made by protein, a big molecule of an integral protein that through which diffusion can take place. So diffusion is not necessarily passing through the lipid bilayer, but diffusion can take place through an integral protein, through a channel protein or a pore of a channel protein. Some of these proteins, they allow the diffusion of water. Water can also diffuse from the place where there's high concentration of water to a place where there's low concentration of water. And this process of diffusion of water has a special name. It's called osmosis. So osmosis is the diffusion of water. It doesn't require energy. Not all the sides of the lipid bilayer allow the passage of water, but sometimes there are some spaces like cracks in the lipid bilayer which allow the passage of water. The remaining part of water will pass through the pores which are present in the membrane, through the membrane pores of the proteins, the protein molecules, through the protein molecules that form the integral proteins. And in this case, those integral proteins that allow the passage of water have a special name. Okay? So they are called aquaporins. So aquaporin is an integral protein that provides a pore for the passage of water. You have to differentiate between diffusion and filtration. Filtration is a process where substances move across the plasma membrane down a pressure gradient. It's not related to concentration gradient, it is the pressure gradient. For example, passage of substances from capillaries where there's high blood pressure inside the capillary bed and there's low pressure in the interstitial or surrounding fluid. So substances will be pushed, will be filtered from the blood to the tissue, even if there's a higher concentration in the tissue, because they are pushed by the pressure. This is something else. It is mentioned here to make a distinction between filtration and diffusion. And again, I repeat, osmosis is a kind of diffusion that is usually used for the diffusion of water. By diffusion, we don't use energy from the cell. The other and the third type of diffusion is called facilitated diffusion, and this is the type of diffusion that requires a transporter protein. That large molecule attached to a transporter protein changes the shape of that molecule and make it fit to pass through a pore. But again, this molecule, this large molecule, can only pass down the concentration gradient, because this is a diffusion. But it's called facilitated because it is facilitated by a transporter protein. Okay. The active transport uses energy, and this is against the concentration gradient. So these are the two main differences between the active and passive diffusion. In passive diffusion, there's no need for the cell to use energy, and usually the passage is down the concentration gradient. Here, the energy is required in the active transport, and the second thing, it is up the concentration gradient. And this is required for certain functions of cells. As we will see in muscle, for example, muscle contraction requires a high amount of calcium. Nerve conduction requires that sodium will be in high concentration in the extracellular compartment, while potassium should be in high concentration in the intracellular compartment. So there will be influx of potassium and outflux of sodium. This requires energy. It is against the concentration gradient, so it is active transport, and at that moment the nerve will conduct what we call an action potential. We're going to talk about this when we deal with the nervous tissue and the nervous system. The other type of transport is the vesicular transport, which is either endocytosis or exocytosis. And the vesicle is a small spherical membranous sac. It buds from the cell membrane. Remember, I mentioned that the cell membrane is so flexible, it allows the formation of these sacs or vesicle, and also it allows that these sacs or vesicles to fuse with the cell membrane without disturbing the structure of the cell membrane. So it's an active process. It always requires energy, and it's either endocytosis or exocytosis. The endocytosis could be either receptor-mediated endocytosis or phagocytosis or pinocytosis or transcytosis. So in the receptor-mediated endocytosis here, there are certain receptors. These receptors are proteins, remember. They are membrane proteins. And one part of the cell membrane contains certain receptors on the outside. On the other side, on the inner side, there's other kind of protein molecules, specifically called clathrin. These are kind of membrane proteins. And the ligand, the substance, will be attached to the receptor protein, and the vesicle will become invaginated and will form the sac. So the sac will be coated with clathrin. Clathrin was on the inner surface, but when the sac forms, the sac will be clathrin coated. So these are called clathrin coated vesicles, and it contains inside them, it contains the receptors and the ligand, and then these will fuse with lysosomes. We're going to talk about lysosomes next time. They are kind of cellular organelles that contain digestive enzymes. These digestive enzymes are going to deal with the ligand that has been entered into the cell, and at the same time it separates it from the receptors, from the clathrin, so that the receptors and the clathrin will return back, will be recycled back to the cell membrane and to be reused again. And the substance that has entered the cell will be acted on by the enzymes. Sometimes these substances are just transported from one side of the membrane to the other side of the membrane, and this is called transcytosis. The other kind of endocytosis that is mentioned here. Pinocytosis, as the name indicates, it is cell drinking. It is like it's only getting the extracellular fluid inside. So here there will be a vesicle is formed, but there is no clathrin, there are no receptors, and no pseudopods. It's only for the fluids. It's entering the extracellular fluid, passed into the cell, and again by transcytosis, this fluid might be transported to the other part of the cell membrane. And this happens, for example, in absorption. In the cells lining the small intestine, for example, by endocytosis, they take the fluid from the outer, from the lumen of the intestine, and then they transfer it to the other side. The other type is the phagocytosis. In phagocytosis, there is a receptor which attach, like the bacteria or the virus, for example. So there is a receptor protein attaching the ligand. Could be a cell sometimes, like a worn out cell, for example, worn out RBC, and these are going to be entered into the cell. But first of all, there will be what we call pseudopodia, like feet, small feet will be formed, as you can see them here, the pseudopodia, and they will take in the receptor and the attached bacteria or the virus, and once they are in the vesicle, they will be attacked by the lysosome. The lysosome contains the digestive enzymes that destroy these viruses or bacteria or the worn out cells.